some of the things I wanted to talk to you today about is what we've learned regarding model selection and what's important in selecting a hepatocyte model for doing your studies, whether they be toxicology, metabolic, or transporter studies for this. So one of the things that we've known for quite a while is that if you want to have an in vivo predictive model for your hepatocyte, you need to have various components, and this includes uptake transporters, so your drug has to get into the hepatocyte. It also has to be able to get out of the hepatocyte, just like what happens in vivo. And of course, metabolism, we've known for years, is important in generation of metabolites that may have a pharmacological effect or a toxicological effect. And more recently, the regulation, we've known a lot about metabolic regulation for many years. It's been highly studied and required in regulatory documents. Much less is known about the regulation of transporter proteins, and I'll show you some examples of that later and why that can be important. And all of these things are working together to generate an intracellular concentration that's in vivo relevant. And the intracellular concentration is critical because that's the driving force for whatever happens inside of the hepatocyte, including hepatotoxicity, inhibition of metabolic enzymes, and also efflux-based transporter interactions. So all these things work together for that. So we realized many years ago that the other thing that was very important is getting the right hepatocytes for your studies. And so one of the things that we developed was our transporter certification program for this. We realized very early on that the commercially available hepatocytes varied a lot in quality and function for this. And so we developed a program that certified these hepatocytes for transporter function, and we used we used clinically relevant probes that covered 6 different transporters on the uptake side and on the canalicular biliary excretion side, and looked at those and measured activity, inhibition potential, intracellular concentration of these probes, all trying to get those to be in vivo relevant. In addition, we looked at the bile acid profile for endogenous bile acids, because hepatocytes, one of their primary functions is to synthesize bile acids. There's 2 primary ones, and then they're conjugated with both glycine and taurine. And we developed conditions for our hepatocytes where you get the right ratios of the various bile acids. And this turns out to be very important in toxicity measurements, because all of those bile acids have differing degrees of hepatotoxicity. So you need the right ratios of the 6 different bile acids, but you also need the right ratios of the glycine and taurine conjugates. And so we've optimized our culture conditions and medias so that they give the correct ratios of all of these bile acids. And then in addition, we do some characterization of phase 2 metabolism. But these transporter certified hepatocytes are kind of top of the line hepatocytes and provide in vivo relevant transporter function for the studies for this. And so we can take these hepatocytes, but the next thing is to put these in an environment that maintains that phenotype. And we know that if you just take these hepatocytes and plate them on a layer of collagen, the system that forms is not polarized. And by that I mean it doesn't have a canalicular domain. It has a basolateral domain, but all of the transporters that would normally be localized on this canalicular domain are internalized, and they're in vesicles and they're not functioning. So hepatocytes that are not polarized have good uptake, So things can get into the cell, but they can't get out, at least onto the canalicular domain. There are basolateral efflux transporters that are present, but all of the canalicular transporters are non-functional for this. And that's why we primarily use two different models. We use the sandwich cultured hepatocyte model and the Hepatopac micropatterned hepatocyte model. And both of these are fully polarized systems so that they have uptake and they form these bile duct here that are separated from the media by tight junctions, and so things that are taken up into the hepatocyte are effluxed into this bile pocket by these canalicular transporter proteins. And so those are expressed, localized, and functioning in these polarized models, very similar to what is happening in vivo. And so this is important, and a really nice example of that is some work that we did a few years ago with Intercept and looking at obeticholic acid. They were developing obeticholic acid and had done all of their preclinical studies, both for metabolism and transport, in this plated hepatocyte model here. And if you remember, that model has uptake function, but no efflux function. So their compound was taken up, and it so happened their compound needed a canalicular efflux transporter to get out of the hepatocyte. And so it was taken up, but it couldn't go anywhere. So it accumulated to very, very high intracellular concentrations. And when they, did some of the toxicity profiles for that, they saw a very significant amount of hepatotoxicity. And unfortunately for them, their anticipated clinical concentrations were starting at 1 micromolar. So it was critical for them, and these in vitro studies actually predicted a lot of potential for hepatotoxicity. Of course, then when they went to the regulatory agencies and they saw this data, they wanted a number of different clinical drug interaction studies. And so Intercept at that time went ahead and did all of these clinical interaction studies, and the net result of that was they were all pretty much negative, saying there weren't any interactions. So of course they were very happy about that and wanted to go ahead, but they I think realized then that they had wasted a lot of money and, more importantly, time in moving their compound ahead. And we got involved in that time in doing some work for a backup compound of theirs, and so they asked us to repeat some of the work with obeticholic acid. And we used our transporter certified hepatocytes and put those in a polarized model that had functional canalicular efflux. And so their compound was taken up and able to be effluxed into the bile by BSEPT, which was a transporter that obeticholic acid required. And then when we did the hepatotoxicity study, we were able to go up 2 orders of magnitude higher in concentration, well covering the anticipated clinical range, and actually correlating now with both the human and animal in vivo studies for that. So it just emphasizes the importance of having a hepatocyte model that has functioning uptake and efflux transporter function in order to generate these in vivo relevant intracellular concentrations for this. So in generating these in vivo relevant intracellular concentrations, I think everyone would agree that it's the unbound intracellular concentration that's important and is a good integrating factor between a lot of these in vitro tests, especially the non-whole cell test and in vivo function. But direct measurement of this intracellular unbound concentration is very difficult and very time consuming. There's mathematical methods to estimate it, but it's very difficult to get a handle around that. And one of the things that you can think about if you use a whole cell system is that the intracellular matrix has a similar binding capacity and characteristics in vitro as in vivo for that. So the things inside the hepatocyte and the binding in there is similar between in vitro and in vivo. And so you may not be able to, it may not be necessary to directly measure the unbound intracellular concentration if you can get the conditions right so that you generate an in vivo relevant total intracellular concentration, because it's going to bind inside the hepatocyte, and that's kind of going to take care of itself for that. So one of the things that we thought about doing is, well, what can we make the extracellular environment more, how can we make that more in vivo-like? And so one of the things we thought about was adding protein to that matrix that we typically do. A lot of the in vitro studies are done in buffers or medias, but what if we added protein to that? And I think people have looked at protein binding in drug development for many, many years. It's usually measured in a separate experiment, and then that number is used to link the free and bound concentrations and to predict what the free concentration would be based on the total concentration. One of the things that's important to remember is that the way we measure protein binding, we measure the extent or how much is bound, but not how tightly it's bound to the protein. You can measure the tightness or the KDs for that, but it's a very involved process that my advisor made me do a lot in graduate school, which people don't do much of anymore. But we're measuring only the extent and not the kinetics. And so one of the things that was noticed over time is that often the uptake of the highly bound compounds would be greater than what would be predicted based on the protein binding for that. And there's been a number of theories over the years, a specific albumin receptor on the surface that increased uptake, some ideas of rate limiting dissociation from the protein or rate limiting diffusion, and various other interactions at the cell surface. And if you want, there's a nice summary article of some of these things that was written by Christine Bowman out of Les Bennett's lab that summarizes a lot of these very, very nicely. But just kind of looking at the extremes of what would happen. So say we have a compound that is very highly bound, so the extent of binding is very high, and it's very tightly bound, so it doesn't come off the protein at a very quick rate. In that case, the active transport is going to be dependent on the unbound concentration, because that's all it's going to see is the unbound concentration. And under those conditions, then the extent of protein binding will correlate very good with hepatic uptake and intracellular concentration. And kind of the other side of things, where you still have a very high extent of protein binding, but it's very loosely bound, so it can come on and off the protein very, very rapidly. So as soon as it's taken up, more disassociates from the protein. And under those conditions, the extent of protein binding doesn't correlate with the free concentration. It correlates better with the total concentration. And to me, this explains when I spent about 20 years at Glaxo and with other companies, and some pharmaceutical companies were very focused on protein binding because they saw this relationship. Others, there didn't appear to be much of a relationship between protein binding and some of the effects they were looking at. So they weren't that interested in protein binding. But I think this kind of explains some of that for that. So under these conditions, for the free drug hypothesis, if hepatic uptake and intracellular concentration are really dependent just on the free concentration, then we should be able to do an experiment with protein or without protein and get the same numbers if we normalize that for the extent of protein binding. And this is true for cellular accumulation, intracellular concentration, and what we were interested in was biliary clearance. So if the free drug hypothesis is working really good, we should be able to, as I said, run the experiment in the presence and absence of protein, generate the same parameters for this. So we looked at this, and the first compound we looked at was Taurocholate. We wanted to make sure that the protein wasn't somehow affecting the physiology of the hepatocytes, and so we looked at carboxydichlorophorocene diacetate, which is taken up by passive diffusion, cleaved, and excreted into these bile pockets. And there was no difference between the study done in the absence of a physiologic concentration of bovine serum albumin and using 4% bovine serum albumin for this. And in addition, when we looked at some of the other parameters and normalized them for the protein binding, we were able to generate essentially the same numbers. The unbound intrinsic clearance was basically the same. The intracellular concentration was the same. The biliary excretion index, which measures the movement of a compound from the inside of the hepatocyte into those bile pockets, that was also the same. And you would expect that to always be the same because putting protein on the outside shouldn't affect something that's happening just inside the hepatocyte, and so we wouldn't expect a change in the biliary excretion index. We then went on to look at other compounds. We started screening compounds, and some of the examples that came up. So Rosuvastatin. So we did the exact same study with Rosuvastatin. Well, again, when we added the BSA, it didn't affect the biliary excretion index for Rosuvastatin, since that's the inside of the cell into the bile pockets. But when we looked at the intracellular concentration or the biliary clearance for that, the intrinsic biliary clearance for Rosuvastatin was greater than what we would predict in the presence of BSA, as was the intracellular concentration. So you would predict, based on the protein binding, that you would get intracellular concentrations of about 3 micromolar. But when you actually did the study and measured it, they were about twofold of that. So there's kind of a disconnect here between that. So something unique is going on with Rosuvastatin for this. And again, we did a similar study with Telmisartan, and I'm just showing Taurocholate here for reference. But when we did this study with Telmisartan in the presence and absence of protein, the actual intracellular concentration was underpredicted by close to 100-fold. So 2 orders of magnitude difference if you did the study in the presence of protein versus doing it in the absence and predicting what that intracellular concentration would be. So, very emphasizing the need or the difference you can see by doing the study in the presence of protein compared to the absence. And we went on to evaluate some more work with Telmisartan. As you know, Telmisartan is an potent inhibitor of BSEPT. When we did this study looking at Taurocholate biliary clearance in the presence of different concentrations of Telmisartan here, you can see that if you just took the IC50 and calculated it based on the dose concentration, you get very different numbers. But if you calculate an IC50 based on the intracellular concentration, these two curves overlap. So you actually get the same number and get a relevant number. And just in green here, I'm just showing the curve for what the predicted intracellular concentration would be. So you could see that if you tried to calculate an IC50 based on those predictive values, you would be way off in your prediction. So using protein in these systems can give you much better predictions of in vivo relevant intracellular concentrations. And this is especially important if you're doing some PK/PD modeling, for instance. It would be very important to capture these types of compounds. And so in addition to selecting the right model, using protein in the media, there's another area that we wanted to consider, and this is, as I mentioned earlier, transporter regulation. So we've known, as I said, for years that P450s can be regulated and induced, but also transporters can be regulated and induced in this. And we know we are using bile acids as an example for this, because the nice thing about bile acids is they're made in the liver, and they're also, the intracellular concentrations are very, very tightly controlled for this. And there's also been a lot of work in recent years looking at the inhibitory effects on that and trying to predict what happens for hepatotoxicity when you inhibit BSEPT. And the kind of historical hypothesis for this is that normally bile acids are taken up by NTCP and effluxed into the bile either by BSEPT or MRP2. And if you inhibit BSEPT, you get this increase in intracellular concentrations of bile acids, leading to toxicity. And so that was the basis for a lot of companies starting to screen for BSEPT inhibition. But as the data began to come out, and this is some publication by Sarah Dawson, who was at AstraZeneca at that time. I'm not sure she's there yet. But they did this correlation between the IC50 for human BSEPT and the different classes of DILI. And you can see here there's a very weak relationship between BSEPT inhibition and cholestatic or different types of hepatotoxicity. There's a lot of false negatives, false positives, and if you look at the predictability of this, it's only about 50%. So that doesn't give you much information about the relevance of BSEPT inhibition. And just the conclusion was using that parameter alone is not going to provide a lot of prediction for that. And why is that? And so Michael Binken was coming out with some of these publications with that, but began talking about adverse outcomes pathways. And really BSEPT inhibition, we're looking over here at the molecular level and trying to predict something all the way over here. And so there's a number of different steps that happen in between that we're just ignoring for that. And so one of the things that we were thinking of, is there a way then that we can add some conditions and develop a system that captures some of the things that are occurring in the middle here and improve that prediction. And so we wanted to look at, we call it the adaptive response, and how the hepatocyte responds to these higher concentrations of bile acids. And when we started doing these studies, we looked at many different transporters. And so when we increased the intracellular concentrations of bile acids, we saw decreased expression of CYP7A1, which is the enzyme responsible for synthesizing these bile acids. And here the different colors are just different lots of human hepatocytes. We use CDCA as our model bile acid, and just increase the intracellular concentrations of that. And we're just looking at messenger RNA expression of some of these transporters. So when we increase the intracellular concentration of these bile acids, we induce BSEP, not surprisingly, because it's trying to get more of the bile acids out. We also looked at other transporters like MRP2, MRP3 and 4, NTCP, but what we found was, in addition to BSEP, the two other transporters that were induced were the two basolateral efflux transporters, OST alpha and OST beta. And we saw a very significant 100-fold induction for these. And, you know, it's really the, when we look at what's happening to the intracellular concentration of these bile acids. So the blue here is with no treatment, and we're measuring just the total bile acid pool of endogenous bile acids. So these are bile acids that are synthesized by the hepatocyte, not the exogenous CDCA that we added, just the endogenous bile acids. And so in the normal state, that's pretty steady, but when you induce them in the CDCA treated, you see the intracellular concentration of these endogenous bile acids drops very rapidly. And that's because you've decreased the synthesis through CYP7A1 and increased the efflux both into the bile and back across the basolateral membrane back into the blood. So you're driving the intracellular concentration down. And that's why it's not, you can add bile acids to this and you don't see hepatotoxicity in this system. And so we looked further in, you know, how does this actually happen for this? So we took these hepatocytes and exposed them to cyclosporin A, which is a very, at 10 micromolar, pretty much shuts down BSEP inhibition. And so when we look at the biliary excretion index of endogenously synthesized glycolic acid, and I picked glycolic acid just because it's the most abundant bile acid in humans, we see that the intracellular, the biliary excretion of GCA decreases very rapidly, and that's because you're inhibiting BSEP, so it can't get out. So what's happening then, since it can't get out, the intracellular concentration of GCA is increasing over time. And at some point, it gets high enough where it actually activates FXR, and we're measuring that by an increase in FGF19. And so once that FXR is activated, it kicks off this whole pathway that I talked about earlier. It inhibits CYP7A1 and induces BSEP, but you have a BSEP inhibitor there, so it's a wash. But it also induces OST alpha and beta, which increases the basolateral efflux of these bile acids. And so we went on from there to look at, you know, we had messenger RNA data for OST alpha and beta and saw increases in that. We then went on to look at changes in protein, and again, saw a very significant increase with when we treated either with CDCA or with obeticholic acid in the protein levels for OST. We did also see a little bit with BSEP, nothing with MRP3 or MRP4 for this. We then went on in this publication with Jeffrey Edwards, and who was at Intercept at the time, and Sen Guo, who was a graduate student at UNC School of Pharmacy, did some molecular modeling, and were able to show that when we treated with CDCA, that we increased the basolateral efflux clearance by about 6-fold with treatment of these bile acids. And also the biliary clearance was also increased. So this is, I think, one of the first examples for transporters as linking changes in messenger RNA with changes in protein with changes in function for that. So very nice body of work there. So we then went on to look more at what happens. We know FXR is activated. Well, what happens if we antagonize that activation of FXR. So here we're showing the activation of FXR by measuring the OST beta messenger RNA. When we inhibit with cyclosporin, we see not much of a change because it's adapted. When we add CDCA, we see more of a change. And then when we put them together, we get a very synergistic response because we blocked efflux, and so we get very, very high concentrations of CDCA. Now when we treat with triglycerin, for example, which is a weak FXR antagonist, we can antagonize that FXR-mediated adaptive response by about 50%. And then we went and looked for the most potent FXR antagonist we could find, and that was DY268. And with that, we can basically eliminate that adaptive response for that. And so that FXR antagonism then prevents the hepatocyte from responding to these high intracellular concentrations of bile acid and can lead to hepatotoxicity for this. So what we think is actually occurring now with hepatotoxicity, especially cholestasis, is we have the normal vectorial flow of bile acids, uptake by NTCP, efflux through B-sept, and if there's now some type of insult where we've blocked B-sept, then the intracellular concentrations of these bile acids will go up to some point, and then it'll reach some new steady state. When you get FXR activation, it'll bring on board this OST alpha and beta, and you'll reach some new steady state level for these bile acids. So it'll have adapted to that initial insult. However, if there's some other secondary insult, or if your compound, in addition to being a B-sept inhibitor, is an FXR antagonist, kind of like triglycerin here, then you have some other things. So you've blocked or reduced this FXR antagonism, and then if it has other effects, either inhibiting the basolateral efflux transporters, either MRP3 or MRP4 or OSTs, that's when you can really run into trouble. And now you've blocked all the ways that the cell has to adapt to that, and you can get this hepatotoxicity. And so we think the cholestatic hepatotoxicity requires multiple mechanisms. B-sept inhibition is probably one, plus maybe basolateral efflux inhibition or FXR antagonism would cause these to occur. And so we integrated all these factors into our C-DILI assay, and now that can be used to, we think, better predict the potential for cholestatic hepatotoxicity. So our C-DILI assay uses the transporter-certified human hepatocytes. It's in a 96-well plate format. You can get it as a kit or a service. We've optimized these culture conditions with various medias, so they kind of sensitize the cells so you can get a response in a 24-hour exposure period. We use our standard culture media just as a control for what we call direct hepatotoxicity. And so we have a positive, negative, and direct toxicity controls for this. And you can use this plate however you want. You can look at 12 compounds at one concentration, or one compound at 12 concentrations, or any permutation of that. It's a 24-hour incubation time. You just measure LDH and ATP, so very rapid turnaround. We use the validation of 50 drugs selected from the Morgan and Dawson papers and the NIH liver tox database we use to rank the compounds. Using this, we increase the accuracy from 50% with B-sept inhibition alone to about 95% using this C-DILI methodology. So I think it's very important. As I mentioned, we use triglycerin as our positive control, cyclosporin as our negative control, because even though cyclosporin is a very potent B-sept inhibitor, the hepatocyte can adapt to that. But with triglyceridone, it's doing multiple things. As I mentioned, triglyceridone and its sulfate metabolite are both very potent B-sept inhibitors. The sulfate metabolite is about 100-fold more potent than triglyceridone, so it's probably responsible for most of the B-sept inhibition in an in vivo situation. And in vitro, it's also generated in this hepatic assay. But as I mentioned, triglyceridone is also a weak FXR antagonist, and it turns out the sulfate conjugate for triglyceridone is also a very potent inhibitor of OST alpha and beta. So you have a compound that is a B-sept inhibitor, potent B-sept inhibitor, an FXR antagonist, and it inhibits OST alpha and beta, basolateral efflux. So it's hitting all the pathways that the hepatocyte has to respond to this initial insult of B-sept inhibition. And so that's why you see a lot of toxicity with that. I mentioned cyclosporin is our negative control. There's other assays, B-sept assays, that use the cyclosporin as a positive control. And it is hepatotoxic, but you have to go to relatively high concentrations inside the cell in order to see that. And so in our study, we looked at the increasing the concentrations of bile acids in the hepatocyte, and then looked at viability just in the absence and presence of cyclosporin. And we did see toxicity, but there's no difference in these curves, indicating, the hepatocyte can adapt to those increased intracellular concentrations. This is just showing some of the data that we typically get from a C-DILI study, just all the control data for that. Again, we do this in our normal culture media and in our sensitization media. We also generally do a concentration that's higher than what the systemic concentration is, because most of these drugs are administered orally, and we're trying to account for the higher portal vein concentrations in that case. So we'll usually go 10 to 20x of the systemic, Cmax or steady state concentration for that. But you can see there's the imatinib, which is our direct toxicity control, and then cyclosporin, there's no difference between the normal media and the sensitization media. And then with triglyceridone, there's no response in the normal media, but a very strong signal in the sensitization media for this. And, we've defined the toxic potential. We have a 200% cutoff for that. We then, for some of the positives from that, we went and looked at what's actually the mechanism behind there, and looking at triglyceridone and DY268, we knew they were FXR antagonists, but we also looked at deferrisox and ketoconazole, and again, both of those are showing changes or decreases in the amount of FGF19, indicating that they're FXR antagonists for that. So you can also, as I mentioned, you can also in the same system then look at the mechanism of why you're seeing that hepatotoxicity. And recently, there's been some work with this assay that identifies conditions where you don't have to have, you can get hepatotoxicity without B-sept inhibition for that. And this is shown with some studies on tyrosine kinase inhibitors. They looked at 6 different ones here, and they proposed a number of mechanisms for that, including B-sept inhibition. But when you actually went and looked at that, the B-sept inhibition wasn't relevant in this case, because you never were able to achieve those intracellular concentrations that were required to inhibit B-sept. And instead, The toxicity, when they looked at the mechanism for this, it was actually due to induction of CYP7A1. And so these tyrosine kinase inhibitors were inducing the synthesis of bile acids. They were increasing NTCP, which was increasing the uptake of these bile acids, and they were decreasing MRP3 function. And so all those things came together to generate high intracellular concentrations of bile acids, leading to hepatotoxicity. So our C-DILI assay is used at different stages. You can use it in the discovery stage to kind of screen a number of compounds, preclinical stage to kind of focus in on concentrations, and then clinically, often your compound is administered with other compounds. So, if your compound is a BSEPT inhibitor alone, it may be clean, but if it's co-administered with an FXR antagonist or a compound that inhibits basolateral efflux, that may be a problem. So you can screen kind of these for the drug interaction potential for that. And you're integrating, all of the acute effects. So this is a metabolically competent system. And you're also integrating the chronic effects for this, that whole adaptive response that I've talked about. And it's really, again, the net effect on this that determines, whether your drug has the cholestatic potential or not. So just to summarize everything, we think studies performed in polarized cell models using transporter-certified hepatocytes that have in vivo relevant transporter function and addition of a physiologic concentration of protein in your media will give you the best opportunity to generate in vivo relevant intracellular concentrations that drive all of those processes that we're interested in inside the hepatocyte and improve prediction not only of hepatotoxicity but induction both of transporters and metabolism, and will integrate the both the acute and chronic regulatory effects. All of this we think will optimize prediction of the in vivo effects and interactions. And I thank you for your time and attention, and be happy to take any questions now or later when we're talking.